In this lecture video, we're going to go ahead and continue talking about chemical messengers, and we are still focusing on membrane-bound mediated responses, and more specifically, we're going to focus on what a G protein linked receptor is. So remember, if we're talking about membrane-mediated responses, we're talking about lipophobic ligands that bind to the receptors. Before we get into any mechanisms, you need to understand the basics about a G protein linked receptor. So you need to understand what is the structure of a G protein. So first off, what does the G stand for? Okay, this G stands for guanosine. This is a nitrogenous base. Within the intracellular fluid, so here's the ICF of this cell, okay, you're looking at three subunits that make up the G protein. These include alpha, beta, and gamma, as you could see there. Notice that you have a GDP, that's what the G guanosine diphosphate is attached to the alpha subunit. That's a very important concept. GDP is a nucleotide like ADP is a nucleotide. Okay, so to activate a G protein, how does it get activated? So first off, your messenger, your ligand, which remember has to be lipophobic, has to bind to the receptor. So remember, we're dealing with slow responses here, so the receptor and the channel are different proteins. So when the ligand binds to the receptor, what happens to happen next? So step 2 then, GDP falls off the alpha subunit. And when GDP falls off the alpha subunit, GTP is added. And when GTP is added, the G protein is now activated. And when it's activated, it's the alpha subunit that slides over. And when it slides over, it will either bump into a channel or it will bump into what's called an amplifier enzyme. I'll show you examples of both of these later. Now, when this G protein is active, this can either alter enzyme activity on the cytoplasmic side of the membrane, or it can alter an ion channel. Okay, so once again, we'll come back to that later. Now, you shouldn't leave your G protein activated, correct? Because if the G protein remains activated, then the target cell keeps making a response, which you don't want the target cell to continue to do that indefinitely, because then you're no longer in homeostasis. Okay, so to inactivate a G protein, the ligand must be removed, or it must fall off the receptor. Remember that binding to a receptor is reversible. The second thing that must happen is that GTP must be hydrolyzed. Remember when we hydrolyzed ATP, you got an ADP and a phosphate? The same is true here. When you hydrolyze GTP, you get a GDP plus a phosphate. So when that happens, then your alpha subunit slides back over and all three subunits are bound once again. So the thing is that you need to remember that G protein linked receptors are an example of a ligand-gated channel. These are slow ligand-gated channels like I mentioned earlier. And so if they're slow, are the receptor and the channel the same protein, or are they two separate proteins? Well, they're two separate proteins. And slow G protein linked receptors are involved with these two mechanisms that we will discuss. There's direct coupling and there's second messenger. And we'll be discussing each of these. So let's look at direct coupling first. So here in direct coupling, again, got to get your bearings. Here's the extracellular fluid in blue. Cytosol is in tan. Here's your ligand. Your messenger is your ligand. So it binds to your receptor. So then when that happens, we know that we activate a G protein. And that's what's going on in step 2. And we mentioned earlier how a G protein is activated. So the GDP falls off the alpha subunit, GTP is bound to the alpha subunit, and the alpha subunit slides over. And here's the thing, in direct coupling, that alpha subunit smacks into a channel. Here's our channel in pink. This channel will either open or or it will close. That's a very important concept. When we talked about fast channels, remember we said they only open. In direct coupling, the channel can open or it may close. Okay, so let's assume that the channel opens. If the channel opens, then you can have ions move into or out of the cell, and then that will change the electrical properties in the cell. So remember that that's one of the functions of a membrane-bound response, is to change the electrical properties inside of the cell. So that's direct. Direct coupling, just think the alpha subunit smacks directly into the channel. Okay, then we have the second messenger system, which is also G protein linked. So in general, so we're going to look at this from a broad perspective first. Second messengers are triggered by the first messenger. That's what the ligand is. And that activates your G protein. Nothing different so far from direct coupling, but now this is where things differ. The G protein then is going to activate the amplifier enzyme. And when the amplifier enzyme is activated, that's going to activate a second messenger. There are different types of second messengers that we'll talk about later. The second messenger then can either activate or inhibit cellular pathways. Okay, so here are the various amplifier enzymes. When you say adenyl cyclase, this is also adenylate cyclase, and guanylyl is also guanylate cyclase, and then there's phospholipase C. And so what adenylate cyclase does is it's going to convert ATP to cyclic AMP. Guanylate cyclase, that amplifier enzyme, will convert GTP to cyclic GMP, and phospholipase C will convert your membrane phospholipids over to IP3 and DAG. These here on the end are your second messengers. Okay, so then let's take a look at the various second messenger systems. So cyclic AMP, cyclic GMP are both nucleotides. So remember what makes up a nucleotide. DAG and IP3 are lipid derived. And then there's calcium, which is an ion. So these are the 5 second messenger systems that we will be discussing throughout this semester. And right now, in a separate video, I will introduce you to the cyclic AMP second messenger system, as well as DAG, IP3, calcium. When we get to phototransduction, or we get to the eye, vision, we will talk about the cyclic GMP second messenger system. So that concludes this video.